please come by and talk. You'll have my contact information in the presentation shortly. And we'll, the facility is basically here to help our users here at Caltech. We're ready to, talk about your research questions and go through anything you want to know about flow cytometry. That's basically why we're here. So we are a multi-user facility. We're made up of, right now, 3 staff members. We're having another staff member join us very soon. And we're basically a repository of flow cytometry expertise, knowledge about how to operate the instruments. And we're there to train you on the instruments, help you operate them. We assist with cell sorting all the time, as well as sometimes with the analyzer work. So please take a look at our website. It's just cellsort.caltech.edu. There's a lot of great information there. So this is the facility currently. I'm the director. We are privileged to have Ellen Rothenberg as our faculty director. She's been a great help and support there. And then, of course, Shelley Diamond, who's the previous facility director, is still very involved and very generous with her time to help out with her expertise that spans over 40 years. So you'll see Jamie Tiarina in the back. She is our kind of expert in all things flow cytometry, but especially small particle work. She will be leaving us shortly for grad school, and we'll miss her dearly, of course. So thank you, Jamie. And then Maddie Adolph is our one of our new staff members, and next to her is Olivia Finney, who will be starting next week. So please come by and talk to us afterward as well. We're happy to help you get things started. So this class is, an intro class. It's a 101 class. We want to cover all things flow cytometry and how they work, right? We want to basically talk about what's going on inside the box so that you're going to be better informed on how to plan your assays, how to execute them, how to do the data analysis afterward. But for those of you who don't know, I guess let's just see a show of hands real quick. How many people have used flow cytometry before? Okay, we have some total newbies. That's great too. But I think this, we wanted to put this together to be helpful for both types of people, right? People that are brand new to it, people that have used it a few times, people that are maybe experts and just looking for some more information. So basic definitions: flow cytometry is a technology that leverages focused flow of particles in suspension to collect optical signals, and we do that in a high throughput manner, right? We pass the cells through a laser that generates scattered light, it generates fluorescence, and we pick those signals up and put them together into a data file that we can then learn a lot of things about your cells or your particles with. Cell sorting is a add-on to that technology, let's say. It was really the beginning. Flow cytometry and cell sorting all happened at the same time, but we now have analyzers that just do flow cytometry, that just analyze cells, and cell sorters that will actually physically separate those cells after they've been analyzed. We'll talk a little bit about that in a bit. All right, so just some basic intro here, and apologies if you've seen these slides from the user group meeting a few months ago, but I think it's really good to rehash these because it really kind of, I think, in my mind, illuminates what flow cytometry is. So a lot of flow cytometry uses fluorescent reporters, and in this case, if you look at a plate stained with a fluorescent reporter, let's say a green fluorescent reporter, we can put that through a plate reader and we can get a measurement of each well on how much expression there is, how much fluorescence. But what flow cytometry does is it takes the cells or particles out of that well and it analyzes them one by one. It's a single cell technology. So instead of just a mean fluorescence of 75.44, we now get a mean fluorescence per particle, okay? And what that does is it shows us distributions of data. It shows us just populations. In this case, a CD3 expression. We see there are some cells that have a lot of CD3 and some cells that have very little CD3. And that can help us start breaking down what's going on in these complex heterogeneous samples. So here's another example of that using Western blots, where you have a kind of baseline, not much expression, and then one that has very high expression, right? And on a Western blot, you see that as not much and a little bit. And here's one in the middle. But when you actually look at the single cells, you can break that down and see that even though this had very high expression, all of that expression was coming from one cell, as opposed to here where you have a mixture and here where there's none. So that's really the power of flow cytometry. And in a lot of ways, it's very similar to the very popular technology that everyone knows about now, single cell RNA-seq. This is kind of where a lot of the data analysis techniques and that sort of thing came from, because we were doing single cell analysis for so long with flow. All right, so these are just some kind of features of flow cytometry, that we can do multiple parameters on each cell. Rather than just one, we can look at 4, 5, 10. Some machines now can do up to 40 different colors at the same time or more. I don't see Caltech doing a lot of that yet, but I'd like to have the capabilities to offer that for those that would be interested. We can also do it in a very high throughput manner. 20,000 cells per second is something we can actually analyze and then physically sort. We can analyze even faster than that if need be. And then it can also be quantitative. Off the shelf, it's not quantitative, but with the right calibration and the right controls, you can get quantitative data out of it. So just a little bit about the progression of flow cytometry. I mean, it all began with imaging and being able to look at something under a scope and look at a field of view and say, this cell is this type of cell, this cell is that type of cell by morphology, let's say. Then adding fluorescence on top of that, you get even more of an ability to tell the different characteristics of the cells apart. Then they started putting them through flowing instruments. So the culture counter is one of the beginnings of flow cytometry. It was an impedance counter. Basically, the cell flowed through an aperture and disrupted a electric field, and that was able to give us information about those cells. But we wanted more, right? And as we got more complex, we took on incorporated the idea of hydrodynamic focusing to focus those cells so that they'll pass through a light source one by one in a really consistent manner. And then we were able to break that out and say, okay, well, I want more than just one color. I want to add multiple colors on there. And that's where instruments as early as the late 80s, early 90s started seeing multiple colors, and chemistry started catching up to give us multiple different fluorescent molecules that we could use as reporters. And now we have all sorts of different capabilities. Spectral flow cytometry is the kind of wave of the future, where instead of just looking at discrete bits of fluorescence, we're looking at the full spectrum for each cell and for each laser and being able to put a lot of information together. So we'll get into that just a little bit in a few slides. So I do want to just point out that flow cytometry and microscopy, flow cytometry and single cell RNA-seq, these are all complementary technologies. They're all going to be beneficial in different ways. And, you know, part of what we do with our consultation kind of intake with the facility is talk to you about your research question and try to figure out, is flow cytometry right or not, right? Maybe microscopy is going to be better. Maybe you can do the same thing for cheaper with single cell RNA-seq, but not likely yet. But that's what we want to talk about and help you kind of figure out how to proceed with your project. All right, so some limitations of flow cytometry. You really need to have your cells in a single cell suspension. There's some maybe caveats to all of these. Maybe if you're looking at aggregates or something like that, they're not in single cell suspension, but we need them to flow through the instrument. So we need them to be below a certain size, let's say, so that we can not clog up the instrument and we can get information about each single cell or particle. The detection limit. We need a certain amount of fluorescent molecule to be able to detect. It varies per fluorophore, but around 40 to 100 fluorescent molecules is kind of where we need to at least be. And that's where for small particles, submicron work, we get into some trouble because we can't load up enough fluorescence to see certain characteristics. It does require expensive instrumentation, although nowadays that's less and less, right? People have flow cytometers in their labs now. Core facilities in flow cytometry really came about because these were, you know, $500,000, $1 million instruments that we couldn't just put in every lab. And they also required really high levels of expertise, technical expertise to operate. That's what the core facility is still there for, but you will hear or see in a lot of your labs small flow cytometers or that sort of thing. We're happy to talk to you about those and help with those, but the core facility is, of course, focused on the instrumentation we have. And then the size of the cells and particles is a limitation, although work like Jamie's been doing has really helped to kind of decrease that number and get down to the much smaller things. And there's a lot of clever little ways that we can get around a lot of the physical limitations as well. And then biosafety is a big issue with cell sorting. We're creating an aerosol with cell sorting, or we can more easily create aerosols. And that can be a big issue if you're working with something that's biohazardous, right? We don't want that to be in the air and being able to infect everybody in the room or anything like that. So we have strategies like containment, biosafety cabinets and things like that. New flow cytometers and cell sorters are chip-based, built on a little microfluidic chip where there's no aerosol. Everything happens in a contained way. So those are going to be kind of the things you'll see coming through in the future. So just to kind of put a pin on that, why flow cytometry? Because it's high throughput, it's high sensitivity. You can multiplex it to have multiple different colors in there, which means you can even do multiple different applications at the same time. And it allows you to explore the single cell data that is in your sample. Why not? Well, maybe you don't have a lot of heterogeneity and you just have a plate reader would be fine for you. That said, there might still be some benefits for flow. You do lose the spatial and contextual information that microscopy gives you. Okay, so that's always something where doing flow is great, but we may also want you to look at them under a scope and see if you can get more relevant data that way. And then the cell is only in the light source for a very short time with flow. So that's where that limitation on how many fluorescent molecules comes in, because the cell is falling through a laser in microseconds. And if that's not enough to create the fluorescent signal that we can detect, then flow might not be the right solution. All right, so examples of flow cytometry data up here. The course and the objective of this is to talk about how this is generated, make sure that you understand what we're looking at here so that you can be inspired for your projects going forward, that you can do better flow in the future by understanding how it works and why making changes in your sample prep, let's say, can result in a better flow cytometry experience. So our objective is to give you the basics and make sure that you understand what's going on inside that box. A lot of the practical details will have to kind of be glossed over here, but that's where we do offer hands-on training on the instrumentation and really get down into what you should do at the machine, how to load a tube and that sort of thing. It's not as interesting to listen to somebody talk about that, I think. It's better to just get in front of the machine and start doing it, and that's what we offer with our training. So also just want to mention that we're planning to do a kind of cycle of a class like this, followed by a user group meeting, followed by a class like this over each quarter. And in between that, we'd love to pepper in some smaller seminars. So if you have any specific applications or specific topics you'd like us to cover, get in touch and let us know. We have a list basically going already of things that we'd like to see either presented by us from the facility or by bringing in vendors to talk about, the vendor experts to talk about their different applications. All right, so now we'll jump into it, right? How does flow cytometry work? Okay, as I mentioned, the goal is to pass particles or cells one by one through a laser and be able to capture the signal from that. So it requires the careful coordination of three different systems: the fluidic system, which delivers your sample to the flow cell; the optic system, which delivers the light source to the flow cell and then picks up the fluorescent and scattered light afterward; and then the electronic system, which quantitates that and then displays it for us on a computer so we can visualize it. And really, all of that comes together at the flow cell. That's kind of the heart of almost every flow cytometer. That's where the light sources from the optical system, the sample flow, and the collection optics all come together in a precise alignment to give us the most optimal signal. So I mentioned hydrodynamic focusing earlier, and that's really one of the most important technologies for the development of flow cytometry, it was a way to allow us to concentrate your sample into a single kind of file flow through the light sources. And the way that works is that we use what's called sheath fluid. Most of you have probably heard of sheath fluid. It can really be anything, but the reason it's called sheath fluid is because it forms a sheath around a core stream of your sample. And by carefully coordinating the pressures of the sheath stream and of your sample stream, we can create a really tight flow of cells through a optimal point where the lasers and the collection optic are all aligned.
Great.
Yep.
Can you take questions?
Yeah, sure. Of course. I should have mentioned.
So if you have the sheath fluid coming in and your input stream is spinning, then it would have to stretch in the Z direction.
Yeah,
so how do you keep the cell centered in Z?
Well, there's so it's because of carefully coordinated pressure, the sheath fluid's moving at a certain velocity through there. At a slightly higher pressure, the sample is injected into the center of that. So by balancing the pressures, you're keeping the cells flowing through the sheath. Does that make sense?
Yeah.
Okay. And that's where we come into, and I'll get to this in a second, but we come into a lot of the problems that can come up when you're working at the flow cytometer, right? If your pressure calibration is slightly off, if there's a clog or bubbles, we can have all sorts of problems where there's backflow, or maybe this stream doesn't go exactly through the right spot. And that's where a lot of the rules that we kind of put in place for the facility that are maybe a downer sometimes, like the cleaning procedures and priming and all of that, really are important to create your stable flow through the right spot in the flow cell. Yep, sure. Yeah, great question. It does not until it's perturbed, right? Or perturbed. Whether there's a clog or when we do cell sorting and it goes through a nozzle, now the sheath fluid and your sample get into a droplet together and they do mix at that point. But before they go through the lasers, they should not be mixing. There's actually a laminar flow to, based on that carefully calibrated pressure differential that keeps them from mixing. At that point, they're mixed, yeah. So we'll get into cell sorting in a little bit, but they form a droplet and they start mixing in that droplet. And then, of course, when they get into your collection container, some of the sheath fluid has come along with the cell. All right, and I do want to just mention that some measurement variability is unavoidable. Some cells will go down the center of the stream. Some cells will go a little off in either direction, X, Y, or Z. And that is something that we calibrate for and we QC for as well. All right, so as I mentioned, we do have a constant rate of flow of the sheath fluid and the sample through the flow cell. The flow cell is a quartz cuvette on most instruments, and that allows the lasers to be aligned not on a much less frequent basis, right? We set up the lasers and they are pointing at their correct spots and don't need to be changed. Earlier instruments, sorry, go ahead. Each of the instruments has slightly different laser configuration, so you can definitely find that on our website, but I'll talk about that in a second as well. Most of our instruments have the 4 lasers pictured here. Our SY3200 does have a UV laser as well. And then the Miltenyi Vibe that we have only has the violet, blue, and yellow, green, no red laser. Go ahead.
I think I have a question to the last part. So for obviously, I'm using the tune system, and they say they have like an acoustic focusing.
Yeah, I'll mention that in a couple slides. I have my own opinions about it, but we'll talk about it in a second. So basically, your speed is consistent. You're pushing the sample and the sheath through at the same speed. And we have this concept of spatially separated lasers, right? They're not collinear. The lasers are not on top of each other. Some instruments do have collinear lasers. Most of the ones we have do not. And that allows us to see a lot more data. We keep the laser lines separate. We keep all the fluorescence generated by each laser line separate, which allows us to have a lot less interference between the different fluorescent signals that we're generating. But if that speed changes, if there is a problem with the flow, there's a clog or a bubble, now we start to get problems with the data because what happens is the timing between each laser is known. We calculate that during QC and we take the data bits from each laser and we put them back together and that becomes your one event, right? As your cell goes through, it hits the violet laser. After a certain amount of time, it hits the blue. All of that is handled by the electronic system and that data is compiled back together. But if that speed is now different from when we did QC, you're going to have the wrong data. And that's where cleaning and priming is so important. So here's where I was going to talk about acoustic focusing, actually. There is, the measurement variability is really the key point here. When we're at low pressure, the low setting on a lot of the machines, we're putting sample into the sheath fluid at a certain amount. And that's creating really good single file kind of cells going through the laser. You can see maybe some are a little off, and that's where the measurement variability comes in. That's why flow data looks like this. We have a CV, right? We're not getting just a line here. We're getting a wider peak because the cells are not going through precisely, right? There's some off a little bit. When we increase the core stream by increasing the speed of your flow, now we have a much wider core stream, right? We're not changing the speed of everything flowing through the flow cell. We're just dumping more volume into it at once. So therefore, your core stream is wider and you'll get a higher CV. You'll actually get a wider peak there, more measurement variability. So that's why, for the most part, we always recommend staying in low setting on all the machines when you're running so you get the most optimal data. The acoustic focusing is a way that the Thermo Fisher got around this with the attuned flow cytometer, where even in higher speeds, they now have an additional focusing on there beyond hydrodynamic focusing, where they use sound waves to keep that core stream tight. And that's why they can claim to be able to run 50,000 events per second without this kind of measurement variability increase. Yeah, go ahead.
We're taking single cell measurements, and so there's an inherent heterogeneity in single cells, which you already showed.
Sure.
But we're also saying that there's a breadth of the distribution due to, as you described, not imperfect focusing extremely. Is there a way to separate out those two sources of variability so that we can really hone in on with the cell-to-cell variability versus the variability that's functioning instrument?
I mean, it really comes down to keeping as tight a core stream as possible to reduce that measurement variability. Measurement variability is coming from a lot of other places as well besides this. It's coming from electronic noise, maybe things in your sheath fluid as well. So there's a lot of different variables that come into it to generate the data. So, having the right controls to be able to see in an unstained sample, let's say, what the measurement variability looks like, and then having your sample that is stained that might have another peak in here, that's kind of where I'll get into controls in a little bit because they are so essential to flow cytometry. You can't tell much about what you're looking at without having had run the proper controls through.
Thanks.
All right, so just some more basics about the fluidic system. As I mentioned, the sheath fluid can pretty much be anything as long as it's aqueous and not going to mix with your samples just immediately, as long as the laminar flow can be maintained. But for the most part, you'll always see it be a PBS-based solution. And that's because on cell sorters, we need the salt in order to create a charge on the droplet. And we'll get into cell sorting in a little bit. But for the most part, you'll see sheath fluid be a PBS or saline-based solution. However, on our Cytoflex S, you'll notice that the sheath fluid we use is just water. All right, so water will work. All we're really counting on the sheath to do on an analyzer is create that hydrodynamic focusing. On a cell sorter, we also have to have it retain charge, and that's where the salt comes in. As I mentioned, it's pressurized. It's going through a constant speed through the flow cell. The pressures that we use can be much higher on cell sorters, so keep that in mind. That's where it can be detrimental to your cells to actually go through a cell sorter. So we'll want to talk a lot about that when you're planning your assays on which nozzle size, which determines the pressure for the most part, we'll want to use. There are gentler cell sorters out there. There are cell sorters that are just meant to go fast. And the faster you want to go, the higher pressure you have to have. So sample delivery does vary quite a bit. On the older instruments, a lot of them were at first syringe driven. Then they started pressurizing the sample as well and using that differential between sheath and sample pressure to create the core stream. But you can use a syringe pump. You can also use a peristaltic pump like our Cytoflex does. The benefits of syringe and peristaltic is that you can also get volumetric data. All right. I always am amazed by the point that a flow cytometer is supposed to count cells, right? And a lot of the time with the cytometers we've had in the last 20 or so years, because the pressure on the sample varies from day to day, we can't get an accurate volume going through the instrument per second. So we can't get an accurate count from them without jumping through a lot of hoops. The peristaltic pump and the syringe-driven systems negate that because you know exactly how much volume you're putting through at what velocity. Okay, so you can get volume data and then be able to get absolute counts. All right, and I've kind of made this point already, but without the regular cleaning that we ask everyone to do, and without priming and making sure that your clogs and bubbles are removed, you are going to run into a lot of problems with your flow data and your flow run. Most of the time, it's going to be a fluidics issue, right? Someone before you didn't clean it properly, or there was air introduced to the system or something like that. That's where most of the problems we see come through with the instruments. So that's always the first troubleshooting step, is to clean and prime it. All right, so that's kind of the fluidics system. We can talk more about it as we go because it's so important that it'll keep coming up. But the optical system is kind of the other end of things, right? The optical system is there to deliver the illumination light to excite your particles or your cells. And that light needs to be at a precise wavelength or a certain wavelength to be able to excite different fluorescent molecules you're putting through the instrument. Each molecule has its own excitation and emission spectra. And then the optical system also collects that scattered laser light and then also the fluorescence generated by the laser hitting the cells and hitting the fluorophores that you may have labeled the cells with. And then we also want to make sure that that laser light is not getting into the fluorescence detectors, because that laser light is so strong, it's going to overwhelm the detectors. So we have to block that and make sure it doesn't get through. All right, so here's this picture of the flow cell again. You see the lasers in spatial separation. For the most part, the excitation sources are lasers. We don't have anything on campus that has an LED or arc lamp excitation, but those things have been done in the past. They may be done again in the future, but for the most part, lasers give us what we want, which is a coherent beam of light that's going to only excite cells in a specific spot and is monochromatic. And then it also it's already, like I said, monochromatic. So it's already at that light wavelength that we need for the excitation. And then going through the quartz cuvettes is going to retain most of the light. So we don't have signal loss due to lasers going through air or anything like that. On some of the cell sorters, we do have stream and air setups where the stream of the sheath stream of the core stream of your sample comes out of a nozzle into the air, and that's where our lasers hit the cells or particles. And those are great systems because they're a lot more flexible, but the problem is you have to have a lot of expertise to set them up. You have to align them every day to make sure that that laser's in the right spot, because every time you turn the stream on, it might be in a slightly different spot, and you need to then dial the laser in so that it's hitting the right spot where you're hitting the core stream, right in front of that collection optic. Okay, and so the spatial separation, as I mentioned, keeps those fluorescent signals separate. So we can then break things down to look at just the fluorescence caused by the violet laser or just the fluorescence caused by the blue laser. All right, and let's say confocal microscopes do that in some ways as well, where they come on sequentially. You'll only have one laser on at a time. Here, we don't have that option because we're falling through space. So we have them all on, but by spatially separating them, we get that same effect. All right, so as far as the signals we're picking up, One of the things we see that you hear a lot about is scattered laser light, forward scatter and side scatter. Okay, this is often the first step to looking at flow data, and forward angle light scatter is really indicates size in a lot of ways, although there's a lot of caveats to that as well. But as you get more scattered light, a higher angle of scattered light, over here, or more, sorry, more light in that angle, that's where you're going to have a bigger forward scatter signal, and that indicates a bigger cell sometimes. So we do have what's called an obscuration bar, which will prevent the laser light from getting into that detector, which really gets rid of all the kind of saturation you'd expect from a laser hitting a detector. And that setup is really important as well. So as a larger cell goes through, you have more scattered light, so you'll get a bigger, a bigger signal here, so it'll move to the right, and you'll be able to separate out large cells from small cells. As you can see here, I put in two different types of experiments. This is a lysed whole blood, and you're able to get very good separation between the monocytes here and the lymphocytes just by the forward scattered light. All right, because the monocytes are bigger, they scatter more light, and that's why the data looks like this. What a lot more people here do is just going to be looking at a cell line, let's say, and there you're able to actually differentiate between your actual cells and maybe dead cells and debris and that sort of thing. And that's where forward scatter and side scatter plots come in as your kind of first step. That was forward scatter. Side scatter is actually at 90 degrees toward where the laser's going. And that gives us information about internal density or granularity, or what processes are inside of your cell. And that's where we kind of call that a structure-dependent signal. And between the two, we can actually generate a morphology picture. All right, so again, this is lysed whole blood, and we can very easily separate out, without fluorescence, lymphocytes, monocytes, and neutrophils or granulocytes. Now, we also can separate out debris down here and dead cells, which would usually appear here, as you can see here, just by this forward side scatter. All right, so that's why it's always, not always, but usually the first step, so that you can get that morphology picture and make sure that you're just looking at your cells. Now, the problem here is that forward scatter breaks down at smaller sizes. So with submicron work, with some smaller particles, you're not going to get as good of a read on your forward scatter. And we have to just depend on side scatter for the most part, and hopefully heavily depend on fluorescence as well. So we can talk more about that if you're going to be doing any small particle work. The forward scatter is really more indicative of refractive index. It just happens to also, correlate with size at the range of mammalian cells.
Do you know what changes in dead cells? So there's a
They usually shrink a little bit. So they shrink, and they also, maybe they start blebbing, or their membranes start folding up a little more. So that's why you see them move down and also up in side scatter. So it's that, effect of that morphology effect, really, that starts to appear in the flow data.
So you'll sometimes have, for both forward scatter and side scatter, like hash K or hash H areas.
Yep. I'll get to that in a couple of slides. Yeah.
So how did you figure out how to color these respective dots in the bottom?
So I'll talk about that in a little bit as well. That's going to be called backgating, but basically by identifying cells with fluorescence on a different plot down the line, you can then display that data on the forward side scatter plot, or any plot really. And then even though we're not seeing that fluorescent data here, we're able to see where those cells show up. Sure. So measurements in flow cytometry. Besides scattered light, of course, we're looking at fluorescence, right? And by just a quick background on fluorescence, most fluorescent molecules have an excitation curve and an emission curve. All right, so a curve of where light shined on them will cause fluorescence, and what that fluorescence will look like when it's emitted. Okay, and of course, fluorescence happens when the light hits the molecule, electrons rise up into a higher energy state, and then when they fall back down, they emit a photon at a specific wavelength. And that difference between where excitation happened and where emission happens is called the Stokes shift. So with fluorescence, you only can get higher wavelength fluorescence than the light source that you excited it with. And basically, we can look at pretty much the entire visual spectrum with a flow cytometer. We do have some limitations in that as you get to the higher wavelengths, you get less energy, so it's not as efficient. So a lot of those dyes work well enough, but if you really need to get a good signal, you usually want to stick to a lower wavelength dye. The problem is at the lower wavelengths, you get a lot more background because a lot more molecules are going to be made to fluoresce by those wavelengths. So it's kind of a balancing act on choosing your panels. And some of you have worked with us to develop a panel, and it is a process where we want to take all of that into account. All right, so spectral viewers are great for determining this. See a show of hands of who's used a spectral viewer before? Okay, they're available. FP-base is a great one, but they're available from vendors as well. Everyone who's selling fluorophores usually has something like this on their website. I think this is the BioLegend one, and this is the BD one. And what they let you do is put up the theoretical data, right, or the tested by someone else data of where does that fluorescence, where is the excitation for it, and where is the emission for it. And, as you can see on the spectra, we can have multiple different fluorophores, and that's where flow cytometry can look at multiple different colors at the same time. So here's just another example. This was a cool old graphic that I found of just a whole bunch of different colors. You can see the violet laser up here. Back then, didn't have a lot of fluorescence molecules excited by them. Here, the blue laser was really the key laser in flow cytometry for a long time. And then the red laser got added on, which allowed things like APC or APC size 7. But what you see on this plot are these kind of gray areas, right? And those are going to be where the bandpass filters are. That's where we're actually picking up the signal from each of those fluorescences. And you can see that it cuts off a lot of the signal. We'll get to that in a little bit. But that's all done using filters. And that's how we tell the machine what colors we're looking for, okay? Each detector doesn't care what light is coming into it. It's going to give you a reading of how much light it sees. But we use them to tell us particularly about specific fluorescence by putting different optical filters in front of them. And we have both the fluorescent and the side scattered light delivered to a detector bank. And each one is restricted by these filters in front of it. And here you can see a long pass filter, a 520 nanometer long pass filter where light goes into it and only the light above 520 comes out. Okay, we have a bunch of different types of filters that we can use. Long pass filters, as I mentioned, short pass filters, which are the opposite, letting only shorter light wavelengths through. And then band pass filters, which have a cutoff on both lower and higher limit, which can really pare the light down a bit. And then we also have the possibility of making any of those three types into a dichroic mirror, which not just cuts the light down, but also reflects light at a very precise angle. In this case, 90-degree angle, or well, 45-degree because it's curved there. But you'll see on different machines that angle could be different, and that's where you have to be really careful when you're ordering filters to work with those machines as well. So we need to simultaneously measure more than one scatter or fluorescence in each cell in some cases, right? Some of you don't. You might just need to look at GFP, but you do want to look at forward and side scatter along with your GFP, maybe a viability dye, so it starts adding up. And that's where we'll use an optical filter detector bank, a detector bank with the proper optical filters in front of it. And here's what was done in the past. This is a fax caliber. Basically, your lasers are going into the flow cell, your sample is coming into the flow cell. Forward scattered light is delivered over here. Side scatter and fluorescence is delivered at 90 degrees up into here. And what you'll see is that you go through several mirrors in a lot of cases to get to a detector. There's a lot of bounces happening and a lot of traveling through different mirrors. What happens is you lose a little light at each of those passing through of a mirror. So modern, and this is another example with the MoFlow doing the same thing with maybe even more bounces, 3 different lasers coming in. The red laser light went over here. This looks like the green laser light, hard to see, and then the blue laser light coming into there. So, we had a lot of sensitivity issues with those older instruments as well. You needed very bright fluorophores. BD actually came out with the LSR2, followed by the LSR rather, then the LSR2, followed by the ARIA cell sorters, using this really clever trick of just bouncing the light for the most part and only passing through a long pass filter followed by a band pass filter. And that really helped save a lot of that light and get better sensitivity. BD is one of the biggest manufacturers of flow cytometers, but this was one of their, I think, biggest contributions beyond just the original cell sorters. So what happens here is that your light from one of the lasers, remember they're spatially separated, so the pickup for the violet laser sends its fluorescent light into here. It hits a long pass filter, which allows only light longer than it through. It then goes through a band pass filter to really pare it down a little more, and then that gets into the detector. Remember, the detector doesn't care what light it sees, but because we're carefully controlling what light gets to it, we can now say, this is the detector where we expect to see BV785. Okay, and same with the rest of these. The light that doesn't pass through this long pass is bounced around. Some more light pairs off here. The rest gets bounced here, more comes through, and so forth. And there we're able to get, on this detector, up to 8 different possible fluorophores coming through. It's actually only set up for 7. But then BD put that on each laser, right? So we have some trigons up here and some octagons, so we can really break down into a lot of different colors on one instrument really easily. All right, does that make sense to everyone? Does anyone know what would happen if we pulled this long pass filter out? So at my old institution, we used to have a lot of different labs coming through that had a lot of different fluorophores. And a lot of the older machines, especially, are very modular. And you could pull filters out and switch them around and make whatever color you wanted work, make it work. But if you did it wrong, or you set it up wrong, or you left it in the wrong state for the next person, they had a lot of issues. Does anyone want to tell me what happens if that one's gone? Yep. Yeah, so for every other detector except really the first 2, because you would still get light here, it just wouldn't bounce. You'd get it cut down by the bandpass filter. So by taking that one little filter out, you now potentially messed up someone's experiment. So that's I guess the point I wanted to make there. Does that make sense? Great. So now, of course, where that was 20 years ago that the LSR2s came out, if not a little more. So we have a lot more state-of-the-art systems in place. The Cytoflex that we have in the lab uses a wavelength division multiplier. And that came from the telecom industry, because fiber optic cables became a really important thing, and being able to tell the different light signals apart in those set off a whole bunch of technological development there. In here we use, oops, I switched over to a different, there we go. These are bandpass filters that are also dichroic. So light hits it, some light goes through, and then it bounces back and forth in this mirror until it goes through one of the bandpass filters, and we're able to pick up the light there with very little light loss. All right, and that's one of the reasons the Cytoflex is so sensitive. And then we also have full spectrum analysis, and that can be done in a variety of different ways. The Cytoflex uses this kind of idea of WDMs. Some of the BD machines use actual old school filters to cut the light up, but really the idea is to deliver the full spectrum of light to the detector so that we can then get the full curve. And that allows us to, first of all, not lose a lot of light. We're taking all of the light rather than just a sliver of it like we would with a bandpass filter. And then it also gives us the full spectral curve so that we can look at potentially unknown things, but then we can also teach the machine what the spectral curve for each fluorophore looks like and then use complicated unmixing software to deconvolute that. So when you have an assay like this, you feed it individual single color controls. This is what GFP looks like. Then in your sample where you have GFP, RFP, M-cherry, BFP, you can then deconvolute them and still tell them apart, right? On a lot of instruments, looking at M-cherry and RFP is tough because they're so close together. They overlap quite a bit. But on the spectral machine, they have different spectra, and we're able to then tell them apart easily. And that greatly expands the number of colors you can use, but also greatly expands the ease of panel design in some ways, because you don't have to worry too much about how much spill there is into each individual detector. You're looking at the full spectrum. All right, so that's kind of the basics of the optical system. We'll come back to it a little bit when we talk about compensation. But I also wanted to mention the kind of bridge between the optical system and the electronic system, which are the detectors. Okay, so each of the detectors here or the detectors here are basically devices to change light into electron flow. Okay, and the original is a PMT, photomultiplier tube, which, the old school ones were vacuum tubes. Light would go in down here. It would then cause an electron cascade that amplified, and that electron kind of flow is correlated to the amount of light that the detector detected, right? We can actually boost the signal by increasing the voltage to those detectors. We'll get to that in a little bit as well. But we also have now things like avalanche photodiodes on our Cytoflex. That's another reason why the Cytoflex has such great sensitivity, because it uses state-of-the-art detectors that are way more efficient than PMTs, especially in some wavelengths. All right, so I'm not going to talk too much about this, but if there's any questions, happy to talk about it. So after the electron flow is picked up from the detector, we're able to measure it. And that's basically the conversion of the light energy into the proportional electronic signal. We can amplify that signal by applying a higher voltage to that detector. And then we get usually digital data out of that on modern instruments. And then, of course, the electronics starts to help provide the data visualization, which all happens in the software as well. So you had asked about the, I believe it was Amy, asked about the dash A, dash H, dash W. That is area, height, and width, and that all comes from the pulse, okay? So as the cells proceed through the laser, this is the laser, this is the sheath, this is the sample flow, and this is the sheath over here, and over here would be the quartz cuvette holding it. As your cell hits the laser, it starts to generate a little bit of electronic flow, a little signal, and then as it goes through the laser, we now start to get the top of the pulse, and then when it passes fully through, we get a full pulse, okay? And that's all detected by detecting the electrons rather than the actual light itself. And we define the pulse by looking at height, area, and width. So pulse height is the first measurement that we used in flow cytometry, which is still around, but has been somewhat replaced by area. Height is just a simple circuit measurement of how much electron flow was there at the maximum, okay? And that's where we get that height measurement from. Area is more robust because we're actually taking the integral under the curve and getting much more of the of that data kind of added into the signal. And then width is a kind of different one where that's looking at the time of flight from the beginning of the pulse to the end of the pulse, okay? And I'll talk about what these are kind of used for. But for the most part, on modern instruments, area is what's used for mammalian cells or those size particles. We start to talk about whether height or area is better for submicron work as well. But for the most part, the default on a lot of the kind of modern instruments is area. Height is still there, and we can look at that. And we can also look at the difference between height and area for some interesting things. But that is the primary one is area. All right, so down here at the bottom, though, we also have this kind of gray line. That's your threshold, okay? And how many of you have adjusted the threshold on a flow cytometer before? Okay, only a couple, because most of the time we use the default, right? The default for the most part is forward scatter, is looking at the threshold in forward scatter, and it's set to a certain amount. That amount is a cutoff. And you can see here pretty clearly, the data just starts to end, okay, on the forward scatter axis. Below there, there's still a ton of stuff. There's still, debris and electronic noise and bubbles and whatever else down here. But we cut that off because we know we're interested in this range of the forward scatter, and we don't want that to overwhelm the signal. There's a lot of that, okay? So we kind of need the threshold in order to really focus down on which data we're interested in. And you can see if we increase the threshold, we start to cut more off, and this is what you don't want. You don't want cells that are potentially of interest to be cut off by the threshold.
So I change my force scatter size signals off the threshold. I kind of play around, like increase or decrease to see what comes in or trial.
Yep.
I find default usually is a bit high.
It depends on the instrument and your sample.
Have things like in the bottom corner, like you're just cutting off the drop just barely, and then you have your data. Or is it better to have higher sensitivity and sort of increase the gain?
I think a lot of those kinds of questions are always going to be, it depends, right? For the most part, I always like to see a little bit of the junk. I like to see a little bit of the debris, because if I don't see that, I don't know where it is. I don't know if there are actual cells of interest down here or not. By being able to see what I assume at this point is debris, I am making sure that I'm set up correctly. And besides adjusting the threshold, you can also adjust the forward scatter sensitivity to bring this down or up. So it's kind of a back and forth between the two to get you the best optimal setup for forward side scatter. All right. So you can also threshold off fluorescent parameters. You can threshold off side scatter. You usually want the threshold to be something that everything in your sample that you're interested in has. And that's why we use scattered light, because everything has a forward scatter signal for the most part. When we get into small micron work, submicron work, sorry, that's where we maybe switch to side scatter because the forward scatter is not giving us as good of a signal, or potentially stain everything, use a dye or a stain that will stain everything in the sample. Maybe it's a lipid stain to stain the membranes or something like that. And then we can threshold on that, a DNA stain potentially as well. All right, so there's a lot of different ways you can use these systems to make sure it suits your data correctly. All right, so then what happens? We have your area, height, and width measurement from each parameter. Potentially, some machines make you opt into the width or the height measurement, like the BD machines. But once you do, you generate a list mode file. All right, and it's really just a spreadsheet, and it'll look very similar to single cell RNA-seq data. It's just a list of cells and what their readings were on each parameter. Okay, and here we're only looking, this is from an old instrument called the MoFlow at the Cyan rather at this time. And that only really had the height parameter. And because of that, you only see, 5 readings here, but each of these readings, forward scatter, side scatter, could have also had a height and a width. So you can all of a sudden get these giant data sets. And that's what, RNA, single cell RNA-seq data blows this out of the water as far as the number of parameters you can look at, but this is very similar to the data produced from those technologies. That's all saved in a .fcs file. That's your raw data file. That's what you're going to bring into an analysis software, and that is really just this. It does have a header, which will have more meta information as well, depending on the software you're using. But that's a standard file defined by ISAC, which is the International Society for the Advancement of Cytometry. And that allows us to be able to use it in almost any flow cytometry data analysis software. There's also, I think I'm getting ahead of myself. I'll come back to that in a second. Go ahead. Say again, sorry.
What are some examples of the parameters of a relevant that you're talking about that can be detected at the instrument?
Well, it's that scattered light that I was talking about earlier. It's the fluorescent light. Is that what you're asking, or?
Yeah, that's like I capture any like from several events. Like it's like I know capture cell size, also fluorescence synthesis, but what else?
Well, on most commercially available instruments, you're pretty limited to those. there's a lot of work going on with kind of integrating other things. I know somebody working on fluorescent lifetime, for instance. Anything that you can measure with that kind of electron pulse, you can build a parameter around that. Yeah, so I mean, we could talk about that later as well if you'd like. But yeah, back to the basics. I mean, that starts to get a little more advanced. But here, we take that data and we can display it as a one parameter histogram. You've seen those probably. The 2-parameter plot that we were looking at earlier. This is now taking, the forward scatter on this axis, but if we put side scatter on the other axis, we can look at a 2-parameter plot. And then, of course, as you get more and more of these columns, you might need to start doing some dimensionality reduction and looking at t-SNEs or UMAPs to really dive into your data a little deeper. And then data scaling is also important. We have with the scattered light usually a linear scale here because we're looking at more linear relationships. But most flow cytometers collect everything in linear now, and then you can convert them into log later so that when you're looking at fluorescence over a much higher dynamic range, you can see everything on the same plot. And then there's also the idea of bi-exponential or hyperlog scaling, where we can insert a linear segment and a 0 and then a negative linear segment onto your scale so that we could condense the low end quite a bit and be able to see even bigger dynamic range. All right, so I'm just going to do a break at this point. So when we come back, we can talk about data analysis and interpretation, some practical concerns. We'll talk about cell sorting, only a little bit, unfortunately. And then we'll go through some of the applications of flow cytometry and then finish up with just more talk about the facility. All right, so let's try to come back by like 11:13 or so. All right, thank you. Yeah. So we have subject dynamics, those are primarily operator assisted. So you book those, you coordinate us, and then we'll apply it for you. It requires one of us.
Oh, I see.
We start talking about going into self-serve at some point, but right now we're for the most part operators. Kind of a reason we can get it to. You need to use it way after hours, something like that, so then we can do training for self-serve. It's very more intense training, so you can start it up, troubleshoot it, and all of that.
I see. So there's no way we can like book it online or something different.
For that, you still do emails. Yeah, and then we'll be able to, but it's in the same calendar. If you click over to sorting at the top, you'll like see the self-serve when they're available, but you're not going to be able to book them without going through us.
I see. That makes sense.
Okay, thank you. Shoot. All right.
So like when I play around with my game, for instance, like I kind of go back and forth between having my negative control cells.
Yeah.
Starting at versus like 0, basically. Versus starting at versus alpha, and then having my experiment cells going up to like 6.
Yeah.
Versus like 0 versus like 1,000, or like 1,000 versus 10,000 or 100 for my negative control and experiment.
All depends on your cells and the experiment. But that's what you're asking, right? How to set that correctly or how to determine that?
Yeah, because I found that one, if you increase the gain, so you see basically as much, you get more false positives. But then if you lower it too much, then you start getting false negatives.
I'll have a slide on it in a little bit, but the real key is to get the best separation between your negative control, your negative cells, and your actual sustained positive cells, right? You want to maximize that so that you can be sure that you have good separation. And then really the controls are most important because that's what's going to tell you where to draw your lines for what's positive or not. But that's a lot of what we'll talk about and look at.
And what do you make of an antibody, or let's say a pair of antibodies? One that gives you nice, homogeneous symmetric peak, and one that gives you 2 peaks. So it's like if the first antibody looks like this, the second antibody looks like this. Or like your experimental cells.
Yeah. I mean, I want to know why that is, right? Is it because antibodies bind them, or did your negative cells look like that as well?
Negative cells.
They're fine. So then, yeah, I mean, what it is this white, oh, the whiteboard's over there. So you're talking about like, one just looks perfect and the other one looks like that.
Yeah, or yeah, something like that.
Over a little
I
bit.
think it's something you use in small bit. Yes,
Or like this.
More like that.
Yeah, yeah, so I mean, this is your intensity, which is amount of antibodies. So if your negative look like this, then this is likely real from your antibody.
So what it looks like is my negative looks like this. My flow cells look like this for one antibody, and then for the other antibody, it looks like this.
Yeah, so there's a lot of troubleshooting that you need to do, right? And a lot of controls that you want to make sure that that this is actually real. First of all, is that actual real antibody staining, or is it background maybe from the antibodies associated with like sticking to all the cells or something like that?
So my negative control cells are cells that suppress my marker, or staining.
Okay, and they look like perfect, yeah, so that's a good control to have, because you're going for non-specific binding, basically. If this is real, I would say this has X number of antibody, and this has slightly more X plus whatever, right? But you have to validate that, okay, you know, that's where cell sorting, you could
Maybe
sort
if
this population, and then you can ask, say, how much is in there versus this population.
maybe if I did this for so let's just sequence, I guess.
That's possible as well, yeah, sorry, I didn't think about that, but we always have a double discriminator in there. Although, if this is a log scale, your doublets should be further out, right? Because they're gonna be twice as fluorescent, at least. So yeah, I wouldn't expect the doublets to quite look like that, but it depends. I mean, if you had more of something like this, then maybe the doublets of these or these guys and something like that, but. You know, yeah, I mean, it'd be something that we'd have to look at the data and explore. Okay, but, yeah, and then also just
So
like
my collaborator gave me these two antibodies, and then they said, "Oh, we use this one because it has higher sigma, it's more stable."
Mhm.
Is what they said.
But yeah, if it's not as ideal of a peak, then that's something to investigate, and I have to go to look at it, take a look at it with you today. How's it going?
I mean, it's not about
Yeah, yeah. Yeah, we'll have to cut down some of the slide. Just, you know, really tough, trying to figure out what to include and what not to include. Yeah, you know, just trying to just do a really broad overview, but it's like, I want to drill down to this, and go on that, but we can do selectively sampling, so each of those things. Oh, great, okay. Okay, perfect.
They just, it was kind of like putting the last night, so I actually didn't see it.
No worries, as long as you're set and there's no, issue there. Yeah, I mean, talk to Matt, I think I would go to some 15-minute, like, a message or say. I wish it was a better orientation, where they told you more about a lot of things, but, it is what it is, they'll have things such as stuff coming up, like benefits and things like that later. They do things like that once a month, so we'll just look into that. And then next week we'll get into all the safety training and all the stuff that you'll take care of. So a lot of e-learning versions. Yeah, I mean, that'll help. We can probably have some people out, I don't know, I was asking you about that. She's not up for herself right now. It wasn't perfect, but now maybe we need to.
Yeah. Right. But you have a 3-year gap.
No, I think it's just standard stuff. So.
Okay.
We're in the process of getting computers out for you. It's important to do something. It's over here. So yeah, she's in stuff.
Yeah, it's very too.
Yeah.
Yeah, I'm one of those people trying to probably bother you. Hey, don't consider it bothering. These terrible panels that I really need, like, take it.
Well, hopefully so, yeah, so good. Follow up on these. Let's go ahead and get started again. If you haven't filled out the sign-in sheet in the front, please try to do that on the way out, just so we have a good record of who is here and kind of keep track of that sort of thing. All right, so everyone's favorite part is how to analyze your cells. And I'm not going to be able to get too deep into it today, just for time, unfortunately, but it's something that we can potentially talk about in a future seminar, do a deep dive on how to do analysis, what you can do with the data analysis, and explore that more. So like I said, let us know if that's something you're interested to hear more about, so that we can make sure we set a seminar up about it. So we have here a bunch of different plots. You know, I talked about the 2-parameter plots. Here we have forward scatter versus side scatter, the same morphology picture we saw earlier, breaking it down into lymphocytes, monocytes, and granulocytes. On a subsequent plot, we have a 1-parameter plot, histogram basically, where we're able to look at just one parameter, in this case CD3. And apologies, this is more immunology heavy, but immunology and flow cytometry kind of came up together. But we can apply, keep in mind, we can apply flow cytometry to pretty much any cell type, pretty much any fluorescent assay that looks at single cells in suspension. So here we're looking at just the lymphocytes. We've put a gate around those lymphocytes, and we're only displaying this population on this plot. We're then able to break that down even further into CD3 positives and CD3 negatives. And then we can display just the CD3 positives on another plot. And this is what a fluorescent plot looks like with immunology, with immune cells, let's say. It's always a lot cleaner than what you'll see with non-immune cells. But we can easily break down double negatives, single positives for CD8, single positives for CD4, and any double positives that might be in there as well. So that's kind of what flow cytometry data looks like. And as I said, you know, those were immune cells. In cell lines or potentially other cell types, you might have more of a morphology picture like this, where we see kind of nice tight cell population with some larger ones. And then your debris down here and dead cells up here. Aggregates would be up here, but we always want to have a doublet discriminator in place to be able to further remove any dead cell doublet cells. All right, so the key point here, though, is that mean fluorescent intensity or median fluorescent intensity indicates expression level. The further up the scale you go, the more of that fluorescent molecule there are in your cells. And if those are attached to an antibody, then the more of that antibody target potentially are in there. Or if it's a fluorescent protein, the more of that fluorescent protein being expressed. So maybe the more expression of that plasmid or something like that. Yep.
So under HU, we don't really get like a good separation. We see like it should be a straight line on the SCSH to area.
We'll come to that in a little bit, but there are a bunch of different methods for looking at double discrimination. But yeah, sometimes it can be difficult to tell them apart. It really depends on the sample type, depends on the instrument sensitivity as well, and what the capabilities of the instrument are. But first, we'll just talk about gating, just in case people are not as familiar with gating. So gates themselves come in all shapes and sizes. You can put an interval gate on a population of a histogram. You can use quadrants on a 2-parameter plot. You can put individual boxes or circles or trapezoids on there to make sure that you get your population. The point I always want to make about gating, though, is that you always want to have some justification for why you included some things versus other things. You always want to have some reasoning there. And a lot of that comes back to having the right controls. If you have the right control to tell you, yes, these are the cells I'm interested in, or yes, this quadrant belongs here rather than up here, that's what you have to depend on to be able to interpret your data correctly. Okay. Gates can be both hierarchical or Boolean, depends on the software that you're using. Hierarchical is going to be a tree kind of setup like that. But Boolean, you can use AND or not gates. Those are kind of on a lot of the older softwares, but a lot of the newer softwares have Boolean gating hidden in there as well in case you need it. You can even gate on the UMAP or t-SNE plots to show where those populations, when they kind of cluster up, where they show up in your actual 2-parameter plots or vice versa. Spectral plots as well. And then I do want to just mention time plots, which a lot of people don't think about. But if we did a class on data analysis, I would always bring those up because they're really important for QC of your actual data run. You'll see all of the cells over time, and if everything's working correctly on your instrument, that should be a very consistent plot, right? All of the data should be coming in randomly, so you should see it be a nice kind of continuum. But if you see dips and valleys in that time data versus another parameter, that might indicate a clog or some kind of uptake issue. And a lot of really high-end data analysis will have a QC step where we look at the time plot, or we have a software algorithm look at the time plot and excise any areas that indicate that the acquisition was interrupted. But ideally, gates are used to identify subsets for finding out what that subpopulation is, but then also to generate statistics about those subpopulations. All right, so here's just an example of some CD25 expression on T cells. Here, by just gating on the lymphocytes, I don't really see the CD25 expression because it's so low. But when I get rid of any of the CD3 negative cells and only gate on CD3, now I can actually see the CD25 because I'm looking at a much smaller subset. All right, does that make sense? Okay. So the different types of statistics, sorry, Amy, go ahead.
It's maybe, separation between like negative and positive.
Of the CD3?
If you're saying cells, or whether or not like antibodies are something like completely good, or
so it really depends. I mean, CD3 positive and negative being so separated here. Sorry, these were CD8 T cells, not just T cells. But that separation is really good. A lot of it has to do with first decision of what antibody is going to be paired with what fluorophore, right? PE is extremely bright and it's going to give you a big jump over your negatives. Then it has to do with instrument setup. What voltage did I decide to set this detector at to give me the best separation? It also has to do with what are these cells? Are these really low background immune cells? They're not moving up at all. If you have a really high autofluorescent cell population, you might have the negatives be more up here. So there's a lot that kind of goes into it on a very sample by sample, case by case basis. But I think the key things are matching up the right antibody with the right fluorophore, or using the right stain if you're just staining, or the right fluorescent protein that has good expression. And then having the instrument set up correctly to give you the most optimal reading. All right. And then, you can't do much about cell backgrounds, right? If your cells are just really autofluorescent, that makes things a lot harder. It's flow cytometry on hard mode, let's say. And there are different methods you could use to kind of get around that by maybe instead of using a lower wavelength, like PE is in the middle somewhere, but still on the lower end, maybe if you used APC or APC size 7, you'd have a lot less autofluorescence in your background. So all of those considerations have to come in order to give you the best data. All right. So, but a little bit more about statistics. I feel like I'm a little out of order here, but that's okay. Statistics are, you know, the actual numbers that we're going to get at the end, probably what you're looking for, right? Unless you're doing a cell sorting experiment where you just want to put a gate around a population and tell us to sort that for you. You want to know about what percentage positives you have potentially, right? And you can use percent parent or percent total or percent grandparent or percent of whatever population you want. But you also may want to know about mean fluorescent intensity, right? This is kind of silly because it's looking at, I say mean fluorescent intensity, but here it's mean scatter intensity, I guess, where along this axis, you have one population showing up here at about, hard to see from this angle, 75 or so. And then you have another population showing up at about 150. Okay, that means that with the forward scattered light, these guys have twice as much signal as these. So does that mean they're twice as big? Potentially. And in this case, these are beads. So yes, these are the doublets of this, and they have twice as much forward scatter signal. Does that make sense? Okay, so we'll see some more data in a little bit with actual fluorescent cells where you can actually look at it is tough to get down to the idea of copy number. I know someone's going to look at that, ask about that for plasmids, say, can I tell how many copies of plasmid this cell had versus this cell? I think that is possible with the right optimization and calibration, but it's going to be a more relative amount for the most part. All right. Median fluorescent intensity, you want to be careful there because both of those are often abbreviated as MFI. So in your papers and such, make sure that you're defining what MFI actually is, what actual statistic you were using. And then we also can get CV. We can get a whole bunch of different statistical numbers based on different populations or comparing the different dots, right? And CV is just how wide that population is. So on the forward scatter, you can see it's a very low CV because they are all manufactured beads that are the same size for the most part. And the reason we have even any CV is mostly that measurement variation we were talking about earlier. All right, does that make sense? Okay, so these statistics can be easily exported, and then you can use them to do your downstream analysis as well. As far as different softwares available to do data analysis, the core has a relationship with FlowJo. We purchase site licenses from them that we can then pass on to our users here at Caltech. It is about $310 a year with the discounts so that you can then have FlowJo on your own computer. There is also a way to get a shared computer for your lab that costs the same amount that multiple people can use. So if you're interested in that and your lab doesn't have it set up already, talk to us and we can work it out with FlowJo to get you up and running. Sorry, go ahead.
Individuals have multiple FlowJo accounts versus on your end, and just to see if we're paying, multiple millions of dollars.
Yeah, I have a access to the portal and see all the licenses from Caltech, so I can easily check that for you. I think your lab just has maybe the one shared lab computer and potentially one other one. I'm not totally sure. I'd have to look. Ah, yes. All right. So they might have quite a bit more licenses. So talk to me later and we'll figure it out. But FlowJo is kind of the premier flow cytometry analysis software out there. It's what a large portion of the market uses. There are a lot of other analysis softwares out there as well, though. FCS Express is kind of the secondary contender there. It's a little more expensive, but you do get some different features. It's more like PowerPoint or Microsoft Word with a ribbon, and it works a little differently than FlowJo. It also has a imaging analysis component that comes with it as well. So you could do some of your imaging analysis there as well. They're actually located right down Colorado and Pasadena as well. So keep that in mind. We do have some free softwares that are available. Things like Summit or WinMDI are pretty old, but they will get the job done. If you're proficient in Python or R, there are a lot of modules available there with good graphical interfaces now as well. So you can try to plug into one of those. A lot of FCS Express and FlowJo can handle plugins from Python or R as well for some higher level analysis. I do want to bring up flowrita.io. Check that out if you'd like. It's a browser-based data analysis solution. It's not going to be as powerful as FlowJo, let's say, but it can get the job done for some basic analysis. Site Expert, which is the software that runs the Cytoflex, is available from the core. It's primarily an acquisition software used to run an instrument, but it has some limited analysis capability, and we can give that to you for free. It is on our SharePoint site for download. It's really nice also because you can build your experiments out in your lab and then bring them with you to the Cytoflex and get started right away. But if you want to do that, talk with us because you need to get our config file for the actual instrument to make it work properly. Okay. Does anyone else know of any other softwares that we could mention?
It's not really software, but one thing I did want to mention is I will quite often just, you can export the raw data just as like a table of numbers.
Yep.
A lot of people will just take those and then plot it themselves using R or like, something else.
Yeah.
That is, you're not making the plots or they're just taking the numbers.
There are some benefits of getting the actual, flow cytometry softwares because then you can gate on subsets and clean things up a little more. Yeah. Stats are the yeah. 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 So I mean, yeah, I mean, it's basically this, right? This is what's inside an FCS file. So you can process that with any software you want that can read this kind of file. Jamie, did you have another?
And if you get the GUI, you can also look at your data. Analytics is one of the more companies, and I know they're offering a free trial, but they're a good company to look into if these options don't work for you for whatever reason you don't do it. There are some older ones too that have been around like Bluesum. I don't go around as often, but that's also an option. And the LOH group, one of the credits is their developed software that you can talk to them about if it's free to use as well.
Yeah, great, thank you. There's just so many now, right? It used to be FlowJo and that was it, but software developers overlapping with flow cytometrists all the time, and people like you said in the Elowitz group developing their own software. There are also very niche softwares like Gemstone and ModFit from Verity Software House. ModFit is really great for cell cycle analysis if you're just doing that. It models the cell cycle phases for you rather than just putting interval gates on it. You can actually get a full modeling. Gemstone is good for differentiation and kind of lineage type analyses as well. So talk with us. I mean, that is really, what, like Jamie will know about some softwares that I don't know about and vice versa. And that's where we can all get together and figure out what's the best software solution for you. And because the FCS file is a standardized file, it should be able to be popped into any of those pieces of software. Or as Amy was saying, it's a list mode file, so you can really analyze it with anything. One of the things I wanted to mention is FlowJo also has a piece of software called SeqGeek available. I'm not sure if has anyone used that. It's for single cell RNA-seq data. it works like FlowJo. It basically reads the single cell RNA-seq data like FlowJo and will make you dot plots and that sort of thing. And then you can have derivable parameters where you can look at individual genes as a parameter, or you could look at gene sets as a parameter as well to try to dive into your data more similar to flow cytometry data. But once you have your software and you have your data available, now you have to think about, how do I want to do this analysis, right? How do I interpret this? A lot of times people just think about percent positive. And sorry, I'm using the same plot here over and over again, even though this is forward scatter, but work with your imagination a little bit. If this was a negative fluorescence, you might want to look at just my positive fluorescence here. All right. That's a very typical application from immunology, right? We're looking at subsets. So we want to look at what percent positive is this marker. And if I could do that on a 2-parameter plot and then drill down to several different markers, I can identify and phenotype cells. One question I want to ask here, though, is how did I know to stop here? How did I know to draw this box like this? Did I just kind of aim for the valley? Maybe, because this isn't real data. But does anyone know how I would do this if I was actually looking at this as real data? Right, exactly. You always have a control, okay? The control is extremely important because that's what's really going to prove to yourself and to reviewers that this is where the positives show up, because I had a negative control that did not have that fluorescence. So now I can actually draw that gate scientifically and objectively, rather than painting a picture like I did when I was putting this together. So that's your most classic application is your gated percent positive. You can also get absolute numbers of that if you have the volumetric data from our analyzers, or if you use cell counting beads that are at a known concentration. When you add them in, you spike them in, you can now get those absolute numbers as well. But the other thing that a lot of people overlook in the immunology circles is the intensity, right? Maybe I'm interested in how much expression there is of this protein or of this target. That's where I'm going to be looking at my mean fluorescent intensity or median fluorescent intensity. But again, it's where you need the controls to have something to compare to, right? You're going to have a gate potentially around this that I know is my positives, and a gate around this, or maybe a separate sample that's my negative that has a known mean at B. And then I have the mean of my actual experimental sample at A. And I can find the MFI-R ratio basically of positives to negatives. All right. And that gives me fold increase. All right. So keep that in mind. But really the point comes back to always have your controls to be able to do this correctly and make sure that you're making that point properly. All right. Does anyone else have any other ways that you'd like to talk about of how to maybe get some information out of your flow data? Well, we can talk about anything later, specific to your type of analysis or your research question. But these are basically the most common ways. So I keep talking about controls. This is showing, the different types of controls that we might need to set up your flow cytometry assay properly. Over here, we have some staining from cells infected with Zika, looking at the E protein. So in condition 1, you have 7.7% infected. Here you have 2% and less. But how do I know where to draw those gates? I have by unstained, or my cells that are infected with the with the Zika. They have E protein there, but I did not add any antibody to the E protein. And now I know where to have my cutoff, and I have a kind of number that I am comfortable with as my background of usually around 0.5% or so that I know in these that's real actual signal. Okay. Other controls that you'll need are instrument setup controls. So you have your negative control here, let's say, but you also need to have a negative control for instrument setup and for compensation if you have multiple colors. And for compensation, you'll also need single color controls. All right, we'll come back to that in a sec. Also gating controls for your fluorescent -1. We'll come back to that in a sec as well, but that's going to be really primarily a gating control, how to know where my actual signal is and not something that's coming from artifacts in the data. Then you might also want to have some antibody binding controls. People have probably heard about isotype controls. They're not in as much demand anymore. It used to be every reviewer for flow cytometry data asked, where's your isotype control? That's not necessarily the case now, but they are useful for being sample prep controls, basically. Am I blocking enough? Am I getting any non-specific binding in there? And that's going to be for primarily antibodies, but you can think about something similar for fluorescent protein work or other types of assays. Positive control is always great to have. Maybe I have a cell line that's overexpressing or something I know is positive. So throwing a positive control in there will help you with your instrument setup, but also validate that your sample works in the wild type, let's say, rather than in one of your conditions. And then, of course, these are the ones that most of you will probably be thinking about as controls, but for us, they're all the way down in the list, right? Your biological controls of what was the treatment, or a treatment, non-treatment control or something like that. So all of these, not all of them necessarily always need to be done, but should always be considered when you're designing your experiments so that you know you have an objective way to interpret your data. All right, the fluorescent -1 controls. I just really love this slide, so I throw it in everything. This is mostly going to be applied to antibody work, immunophenotyping. But what happens is when you have multiple colors in a sample, you have interactions between the colors caused by compensation artifacts that are pretty much unavoidable. And in here, if you use just your isotype control or your negative control to set a gate for your PE, I'm in a 3-color system with FITC, PE, and PE Sci5. When I come over here, I look at it and I can tell right away that the gap between this population and this population is way up here, but my isotype control or my unstained control hasn't identified that properly, right? That's where a fluorescent -1 control comes in, where this one is stained with just your FITC and just your PE Sci5, and you can see that it puts the gate right in the correct spot, right? It does not have any PE antibody in there, so therefore any of the compensation issues or whatever don't confuse us because we're looking at something without PE. So anything that is PE positive is going to stain above that. Does that make sense? Go ahead. Yes. So yeah, we'll talk about compensation in a second. But yeah, because of the different spectral overlap between the multiple colors and the way that we're compensating for that, which is what compensation is, it causes an artifact that causes this. Okay, so having your FMO control is the right way to set those gates. And really the CD4 is a bad example because you have a great separation already. You can see where that should be. It's a bad example because you probably wouldn't bother doing an FMO for this because you can tell where that gate needs to be, but it's a good example because you can see clearly that the FMO worked. Yes.
Something that you would do where you would subtract one color or one dye, like each dye, like once.
Yeah,
So
yeah.
then, like for this, right, you would have no PE and then a no FITC and then a no Sci5 PE.
Correct. And when you're talking about a 40-color experiment, yeah.
Very sample, for instance, then you have to rely totally on your like compensation, like unmixing.
This is going to solve a different problem from your compensation controls and from doing compensation. This is assuming compensation has been done. So yeah, I mean, it does get really tough because if you are sample limited, how do you make all those controls? It also gets very expensive. You're using a dose of antibody for each of, a dose of each antibody for each of the controls -1. Sorry, go ahead and we'll talk more about it.
Basically need those separate measures for the side and then together.
Yep.
Each time you run an experiment, you would run all of those together.
You would run each single color control so that you can kind of teach the analysis, worksheet what each individual one looks like, and then use compensation to eliminate any of the spillover between the different colors. So my GFP spilling into my RFP, I can eliminate that using just the GFP control. My RFP spilling into my GFP, I need an RFP only control to eliminate that. And that makes it really tough when you're talking about cell lines. Sure, I mean, you'd have to keep each of those different cell lines in culture plus a negative control in culture. But when we start talking about mice or other animals, that gets even more complicated. So that's what, you know, our consultation will kind of address with you is how are we going to figure out what the best control is and what compromises do we have to make? You know, at some point you can't, you may not be able to bring all the controls you need to really have an optimal experiment, but we need to try to get as close to that as possible. And that's where, you know, some people will use compensation beads, which I don't touch on here, but we can talk about, or they'll use frozen down aliquots of cells that, you know, rather than keeping them in culture or rather than sacrificing another animal, you do that once and then you have some that you can thaw out each time. There are strategies there, but none of them are ideal as using, you know, the cells cultured at the same time or something like that. All right, so this again applies mostly to immunophenotyping, but it does have lessons in there that apply to everything about how to have the right control to make sure that you're objectively gating things properly. All right, and then I did want to just talk a little bit about quantitative flow cytometry. It's something that a lot more people are talking about these days. And you know that the kind of problem here is, you know, we have these infected cells that have a GFP positive readout for infection, and you have some that are way up here at almost 10 to the 5, and you have some that are down here at 10 to the 3. All right, and that means that the ones down at 10 to the 3 just have less GFP per cell. Okay, so how do we relate that to actual amounts of GFP? All right, can I say there's X amount of GFP here and X amount and 4X amount of GFP there, or 10X or whatever it is? That's where quantitative flow cytometry comes in with calibrating the instrument properly using either beads or other particles that have known amounts of each of those fluorescents. Okay, so if you have a bead that has this bead has 1,000 copies of GFP, and this one has 10,000. Now I can plot that out, and I can then interpolate my actual GFP data into that and get some real numbers on how many copies each one has. All right, that's not necessary for everybody. Some people really just want to know, okay, this has 5x of this or something like that. But if it is something that you do need to know and you're interested in, talk with us. There are strategies to get you to the point where it can actually be quantitative. All right, and here are some of those beads. You can see you have I think it's mean equivalent of PE and mean, yeah, so 770 mean equivalent for 400, and you would know the values for each of these, and you could pop in your means from your actual sample and get some readouts on how many copies of PE are actually in there. As you get more exotic with your fluorophores, these are harder to come by, or you have to make them yourself, which can have its own challenges and issues introduced as well. So most of the more common fluorophores have these available from either Spherotech or Bangs Labs, which are the big bead vendors for flow cytometry. So there's an idea of, you know, using actual PE to give you an actual equivalent, or using something that you can equate to PE with a totally different fluorophore, which has some more actual caveats attached to it, but it is definitely possible. All right, and I did mention counting cells earlier, so I can kind of skip through this one, but we have of the Vibe and the Cytoflex both have the ability to do volumetric counting, so you can get absolute numbers there. With the cell sorters, you will have to actually spike in counting beads of a known concentration and a known volume so that you know how many beads are in there and be able to back calculate the concentration of your cells or particles. So we talked about this a little bit on how to set voltages. It is something that we could probably have a whole little mini talk about, but ideally what you're looking for is the best spread. This is a bunch of concatenated files from different voltage settings. for something that has a positive and a negative expression on this UV laser color. And you can see if I have my voltage set too low at around on this machine 200, I don't really see any separation between positive and negative. Okay, but as I increase my voltage, I start to see better separation. And as I start to increase too far, now my negative population starts to spread out, and my negative population actually starts to increase as well. So this is something called a filtration, which you can do at the beginning of your assay development to figure out where the best voltage is probably going to be for your actual experiment. There are then ways to, sorry, go ahead. Yeah, sorry. So the voltage is actually the voltage applied to the detector. So as you increase the voltage, the detector becomes more sensitive. More electrons are going to flow, and it amplifies the signal in some ways. On the Cytoflex, it's actually called the gain because we don't actually have a voltage on APDs. It's just amplifying the signal.
Because they're the way you set everything parameters. And you don't want to.
Yep.
Sounds like here what you're doing is just trying to look for the highest dynamic range between negative and positive, and not within the distribution of the signal in your sample. Expression, right? Like you have like the tissue or like you're dissociating cells, some may be high, some may be low. If I turn on my detector, they're all high.
Right. And that's what's happening here, for instance, right? If you could go up even further, you'd eventually saturate out the detector. So yeah, I mean, and this is where if you don't have a positive negative in the same control, your negative control comes in that you have prepared separately, right? But yeah, you want that best separation between your positive and negative to know that you can identify the positives correctly.
The correct way to do this, like you said, I have positive, usually negative, as a reference, and you just kind of.
You can do it in the systematic way of checking voltage every 50 volts or 100 volts or something like that and figure out where the best setting is. But it could be different. It's going to be different from instrument to instrument as well. Yeah.
How reproducible?
Say again?
How reproducible once you, like if you have stable lines, is it something because if you're not cells in the dish that are easy to come by, like can you do a bunch of preliminary experiments and say, okay, I'm confident, I know there's some variation, but these are my segments?
Yeah, I mean, you can do this with beads, let's say, but it's not going to perfectly apply to your cells. So doing it with your actual sample is the ideal way to do it. But we can get a kind of head start by using other particles to do this kind of thing with. But yeah, being objective with it is important, and then the consistency is going to depend on how consistent your cells are expressing those things. One of the things that are nice to include would be a landmark of some sort, right? A bead that is at a known fluorescence every time, so that you just take that out of the fridge and put that up, and now I know that my bead showed up here, and for some reason when I put my sample up, it's showing up here. I can maybe turn it down to try to keep that consistent. And there are ways in the different softwares, especially the FACSDiva software from BD, where you can relate that to the QC procedure that went on the instrument earlier in the day or right before you ran, so that you can kind of standardize from run to run.
Yeah, like saying the BI is pretty consistent across your entire population.
That would be interesting. It's a good idea, an interesting landmark. Your cells would have to be fixed and permeabilized so the DAPI can get into the DNA. So if you're using live cells, that wouldn't work. But yeah, I think that would actually be an interesting landmark to have. Yeah.
How critical is it? Because when I look at it, anything between 300 and 650, kind of like gives me those other ones.
Sure. So it's going to become more important the more colors you have and the more compensation interactions you have, because You start to lose low end sensitivity with more compensation. It does start to look more like this just by doing compensation. So you want the best separation so that you have some resolution to lose, basically. If you're only doing a couple colors and you just care about what percentage are positive, no, you don't have to do the ideal flow cytometry experiment. You can just look at this, set the voltage so that you get good separation and know what the percentage is. But it's when things start to get more complicated or you want to be sure you're not missing any low end populations, that's where you want to be able to optimize. Okay. All right. So do want to mention backgating. I talked about that a little bit. I'm going to go a little faster because we're running low on time. Backgating is great to light up the populations for the forward side scatter, because unlike with fluorescence, forward side scatter is a much more subjective and difficult kind of decision to make on what my settings should be to get best forward side scatter. With lymphocytes, with lysed blood, lysed whole blood, it's easy because you have landmarks. You know there's going to be granulocytes. You know there's going to be monocytes. I want to set it so I can see all three of those if I'm interested in them. But if I don't know what I'm looking at here, this looks like a mess. Are these my cells? Are my cells down here? I don't know. You can use fluorescent signal to actually backgate and light up the population. So you can go back and forth. And that's really one of the most powerful things about flow cytometry data is that when you come off the instrument, as long as your voltages were set well, you can go back in and reanalyze and try different gating strategies. Start with forward side scatter. Maybe that doesn't look good. Start with fluorescence. Start with a particular color that you know identifies your cells. And backgating allows you to then see that on other plots like the forward side scatter plot. Does that make sense? All right, so I've got a bunch more slides, but we're running short on time, so I'm going to go through them pretty quick, but stop me if we want to talk about anything. I don't know if someone has this room after us. So compensation, we talked about the detectors earlier. You have all the light coming in, we cut the light up, and we have just the PE light coming into this detector. But some times when you have multiple colors, some of the light from, let's say, AF488 can get into that PE detector. It emits in that same spot where my detector, my optical filter is cutting the light out for PE. So compensation is about removing that from this detector mathematically. Okay, and we do that with the single color controls to make sure that when we put that single color control up for FITSI or for AF488, let's say, we know that it shows up in PE a bit, and compensation is bringing that down to only show up where it's supposed to be in AF488. All right, and we do that by lining the means up, right? I know it's compensated correctly because the mean of the negative is equal to the mean of the positive. I have a nice line parallel to the axis here. Okay, and that's why single color controls are so important. We need to be able to pull that spillover out of each of the single color controls and then apply those settings to my fully stained sample. So in my fully stained sample, I know that these double positives are truly double positive and not caused by some spillover of one of my other colors. Okay? Yep.
What is the advantage of like compensating rather than just increasing the gauge?
Rather than just putting a gate in like this.
It's not a straight line, right? It's like an exponential, or like how do you treat this?
Yeah, I mean, I guess I can show you in here, based on your voltage settings as well, you can have different compensations necessary, right? So if my sensitivity of one of my detectors is tuned up too high, now I'm getting a lot more spillover. Okay, and things start to really condense and crunch down when you have a lot of spill like that. And then if you think about having multiple colors, 4, 5, 10, 12, 40 colors, the data starts to really suffer. You're also losing resolution on the other detector by having the negative be way up here, right? Because I now no longer have resolution on my other channel, on my APC Psi7 channel, because my negative is starting too high. And that's where compensating brings it down so that you have, you rescue some of that resolution. We have what's called the spread effect, which I don't get into much here, but when this is compensated correctly, instead of just moving straight over, sometimes it trumpets out a little bit. So that loss of resolution is something you can't really avoid. And a lot of that comes back to picking the correct colors and making sure that you're doing a lot of that optimizing and legwork at the beginning of your assay, where you have different colors that don't interact as much. All right, I mentioned doublets before. I think someone had asked about them as well. Here we're looking at area versus width. That's one way to look at doublets, where you have higher width from that pulse. Remember, the higher width but same area is often going to be a doublet. So we can use the forward scatter or side scatter width versus area to remove those. Other machines and other companies started training people on height versus area as well. Ideally, your height and area should be proportional and you should have a nice 45-degree angle. But as a doublet goes through, it actually ends up having more area than height, and that's why it appears down here. So we can then exclude those as doublets. Okay. And the point over here is that you want to get rid of doublets because, especially for cell sorting, if you have a negative attached to a positive, it has the exact same fluorescence, or pretty much the exact fluorescence, as a single positive. And that can really cross-contaminate your sort, because now you're getting negatives even though you didn't want to. All right, so I was going to talk a bit about cell sorting. I'm going to just kind of keep it to 1 or 2 slides here, because I do want to get to the end. But the idea is, for any flow cytometry assay that we're setting up on a cytometer, we can sort that as well, right? We can physically isolate those. So someone had a question earlier about a peak where they had one peak here, but then a little shoulder, and whether that was real or not, or what was that. One of the ways you can address that, other than changing up your sample prep, is to actually sort those out separately and analyze them in a downstream assay. Okay, because we can physically isolate them. You can bring them to single cell sequencing or a Western blot, or look at them under the scope and see what the difference is. Run a PCR. We're able to sort up to 4 populations simultaneously on our instruments. We can also do single cell sorting into well plates, so you can generate clonal lines, or do some single cell analysis either downstream with proteomics or genomics. And we talked about this at the beginning, but you do get a mixture of your sheath and your sample buffer in this process, and you're going to get a few nanoliters of the sheath fluid along with your cell in each droplet if you're doing single cells. But if you're doing a bulk sort, that volume starts to add up, so keep that in mind as well. All right, so I'm gonna skip a lot of this, but just give you the basics, where we're basically using a nozzle to generate a stream, and we're applying a vibration to that nozzle that breaks that stream up into droplets. And then, by knowing which droplet has a cell in it that you actually want to recover, we can apply a charge to that droplet, which then go through an electric field set up by 2 high voltage plates. And because there's a charge on the droplet of the cell you want, it gets flung, and you can do up to 4 different charges, either like double positive, positive, or double negative, or negative. You can spread that over 4 different collection tubes at the bottom. All right, so that's the quick and dirty cell sorting. Talk to us. We're happy to talk more about it, and we can probably have another seminar just on cell sorting even. So the important thing about cell sorting is that we know the distance between the lasers and that last attached droplet. So that's how we can charge it. We know that when the cell or the droplet travels from the lasers where we knew where it was down to that last attached droplet, that's called the drop delay, and that's how long we wait to actually charge the cell. So if anything shifts or changes during the cell sorting process, now that calibration is out the window, and that's where we're not going to get a good result at the end of your sort. So keeping your cells strained and avoiding clogs and avoiding running the tube dry is something we really strive to do to make sure that we don't get a bad sort. All right. I did want to mention large particle sorting. Talk to us if you're interested in that. We are potentially going to be acquiring an instrument that will be able to do that. We're talking up to about 700 or 1,000 microns. And it works a slightly different mechanism than our usual sorting, and it's a lot slower, but you're able to sort things like seeds or embryos or organoids and that sort of thing. So usually the cutoff for the machines we have in-house is more like 20, 25 microns because the cells start to get too big for the system. All right, I'm going to skip through a lot of these applications. I know some people wanted to hear more about the applications, but I'm going to have this actually put up on our website. This is a list of different possible applications, and it's not an exhaustive list, but these are all things we can do with flow cytometry. And the take home is that we can do them in high throughput and also physically isolate them. And we can also multiplex different types of applications as long as the prep doesn't interfere with each other. All right, so here's a couple. I'm just going to run through them real quick. We can look at cell cycle. We can get really good distinction between G1, G2, and S phase. We can look at apoptosis by looking at sub-G1, by looking at expression of caspases or mitochondrial potentials. A lot of these are different assays that look at apoptosis that happen at different parts of the apoptotic window. These are the assays down here at the bottom that we can actually do with flow. Here we can look at proliferation, where we put a dye into the cells, and as it divides, you get half as much dye each time. So we can actually do a generational analysis of cells as they're responding to whatever treatment you gave them. RNA flow is something that is really interesting, where we can actually look at cell surface staining for whatever proteins on the cell surface you're looking for, but also intracellularly look at RNA expression. Okay, and so you can do, I believe, with the new kits up to 4 at the same time. So instead of, I've had some users come and they'll sort, and then they'll want to do a qPCR on their sorted cells for a specific target. But with something like this, you don't need to sort. You can just do all of that right on the analyzer by actually looking at the transcripts as you're flowing. This one is one of my favorites. It's the multiplex, basically ELISA. Instead of doing this in a dish, each bead that you put in there has capture antibody on it, and you're doing a sandwich ELISA on the bead. But you can then mix beads that have different targets or antibodies for different targets together, so that instead of doing 8 ELISAs, you do this all in one tube and run it on the flow cytometer. All right, and it also works with a lot less sample volume than a classic ELISA. Some of the technologies go up to about 50 different analytes at the same time, so it's really powerful. But when you get up to 50 analytes, you start running into problems with this one needs to be diluted 1 to 200, this one needs to be diluted 1 to 500, and it becomes a little complicated, but you do get a lot of data out of it. And then small particle flow cytometry, talk to us about it, talk to Jamie. This is something that's a big kind of open frontier in flow cytometry, because we're going to be able to look at all sorts of different submicron particles in a high throughput way that is going to be a lot easier to prep for than doing electron microscopy or something like that. And then as well, Jamie's done a lot of great work being able to sort those small micron, I keep saying small microns, submicron particles. And, you know, this is where I was talking about the refractive index and forward scatter breaking down as you get smaller. It is really about the refractive index. And down here, you can see, you know, beads might have one refractive index, but the same size organic particle might have a very different refractive index. So a lot of people are looking at how to standardize and use beads or other maybe viruses as a calibration particle to help us figure out what the sizes are. So like I said, these are just some of the applications of flow cytometry. Get in touch with us, cellsort@caltech.edu, and we can, we can talk about your research project and how we can apply it, you know, any of these things or multiple of these things to your samples. Okay, so I talked about why not flow cytometry already earlier. I did talk about multiplexing and needing the same sample prep in there. And I also mentioned this, that we want to do these quarterly events, do this class over again. I'll try to tighten it up a little bit so we can finish on time. And then also have kind of smaller events peppered in there as well. So get in touch with us about what you want to hear about, whether from us or from an expert from one of the vendors, or I don't think we can invite experts from other institutes, but that's always a possibility as well. So working with the facility, this is kind of down to the real practical how to do this. Get in touch with us with the email address. That's really the key. Email cellsort@caltech.edu. We have a biosafety questionnaire on our website that you can fill out that gives us a bunch of background on what your research question is. We can then study up and get together with you in a meeting and put together some ideas on, you know, how best to address your question, or if you already kind of know what you're doing, how to optimize and make sure that you're going to get the right data out of it. And as Shelley says, there are no stupid questions. So please reach out to us and talk to us. That's what we're here for. Our scheduler is up and running for the analyzers. These are our two analyzers, the Cytoflex S and the MaxQuant Vibe. As I mentioned to some of you earlier, the Vibe is at the kind of end of its life. So we're thinking about how to replace that and what to replace it with. So if you have particular needs that don't seem to be met by the current analyzers, if you need a spectral machine or you need an imaging flow cytometer, which is another kind of frontier of flow cytometry right now, talk with us, and those might be possibilities for replacement. We do have 3 cell sorters, the Fax Fusion, the SY3200, the Cytoflex SRT. Those are all available for operator-assisted use, where you book with us and then we can sort for you. On a very case-by-case basis, requiring a lot of training, we do offer self-serve sorting if that's something you need for after-hours work, or you know, for whatever other reason you might need to be doing it on your own. All right, and here's our staff. Please talk with us. Talk with Maddie and Olivia. They're our new staff members right here. So say hi. And yeah, so lastly, feedback is essential. Let us know really across all aspects of the core facility. If you had a bad experience, a bad sort, a bad run, the instrument wasn't in good shape when you got there or something like that, let us know so we can actually address that. We've had a couple situations where there was a problem with an analyzer and no one told us, and we didn't find out till the next day or something like that. So it impacted a couple other people. Tell us right away. That way we can make sure to get to it. And then in general, you know, anything you'd like to see the facility doing, offering the different types of seminars I'm talking about, or anything like that, get in touch and give us that feedback. So my email is down here as well, but the cell sort email will get to me as well as all of the staff. All right, and that's it. So if you have any questions, I'm happy to stick around later, but again, I don't know about the room, so we'll have to see. All right, thank you.